Hi everyone, I'm Hannah Solomons, a postdoc in the Hair Lab at Duke University. We are proposing a comparative study of working memory and are looking for a variety of species to participate. Specifically, we're looking for insects and similar arthropods, all kinds of vertebrates, and cephalopods. We are proposing this study as part of a larger project looking at the major brain transitions hypothesis. So the hypothesis is that major evolutionary transitions in brain structure will be reflected by major transitions in cognition. And for this study, we're specifically looking at the transition between recurrent brain structure, like that is found with insects and other arthropods, to laminated brain structures, which are found in vertebrates and cephalopods. Our prediction is that species with laminated brain structures will be able to remember the hiding location of an object for longer than those with recurrent brain structures because of the way that these brains process information. So to fully test this hypothesis, we need a large-scale phylogenetic study across many species with each of those brain types, and that is where our call for collaborators comes in. So the details of the experimental setup for this working memory test are going to vary a lot based on the species. But within that variation, there's some important elements of the test that we'll need to achieve for each species. So in a moment, I'm going to show some video examples of how we did this test with dogs here at Duke. And when you contact us to participate, we will get on the phone and discuss how we can make the test work with your species. But here's an overview of how the test is going to work. So subjects are going to be watching a target, which would usually be food or a treat, be hidden in one of three hiding places. Then there's going to be a delay of either less than 1 second, 15 seconds, or 30 seconds. And during that delay, the hiding locations will be blocked from the subject's view using an occluder of some kind. After that delay time is up, the occluder is removed so the subject can see all the hiding places again, and then they're allowed to enter the area and make a choice of hiding location. How they make the choice again can vary, but usually it would be by touching that location. So if they choose the correct location, they will be rewarded, and if they do not choose the correct location, we just move on to the next trial. So before we get started with the test trials, we are going to have all the subjects complete some warm-ups. So there will be 4 stages of familiarization that will help the subject incrementally get closer and closer to the test procedure so that they're comfortable with that testing protocol. So these stages of warm-ups are described in detail in the methods document. The final stage of warm-ups is identical to the test trials, except that there's no delay and no occluder. So they just see the treat being hidden and then immediately get to make their choice. So we'll show a video of that right now.
Puppy, look. Puppy, look. Okay. Choice. Yes, good boy. Are you ready for the video?
Subjects will be required to get 4 out of a sliding window of 5 of these final warm-up trials correct in order to move on to the testing phase. This ensures that they're willing and able to choose the location of the target when there's no delay. The test trials will be the same as those final warm-ups, except that in the test trials, after the target is hidden, there will be a delay of either less than 1 second, 15 seconds, or 30 seconds. During that delay, all 3 hiding locations will be blocked from the subject's sight by an occluder. Here's an example video of a less than 1 second delay. You'll see that with the dogs, we used a curtain as the occluder to block the sight of the hiding locations during that delay. For the less than 1 second delay, the occluder is placed for just a moment and then immediately removed.
Trial 3, 0 seconds. Puppy, look. Puppy, look. Okay. Choice. Yes, good boy. Good boy, boy. Good job, you did it.
And here's an example of a trial with a 15-second delay. Remember that there will also be trials with 30-second delays. Please note that during the delays, the subject is free to move around the space on the other side as they please.
Again. Okay, trial 4, 15 seconds. Puppy, look. Puppy, look. Okay. Yes, there you go. Good job, honey. Good job.
Thank you so much for your interest in our study, and we hope that if you have species that are relevant here, you will choose to participate. If you want to participate or just find out more about whether you think this study can work well with the species that you have, please click on the green Request to Join button here on our PhyloPsy project page. Thank you so much, and we look forward to chatting with all of you.